fairly, and rather than just have me have just a recorded lecture. So this is our first, and sort of what I like to think of as kind of the second chunk of this class, and it's an important chunk in that we're moving beyond just thinking about kind of the host picture of what are we doing, how are we sort of fending off pathogens, and moving into more of what are they doing, how are they initiating this interaction. And so where I want to begin is talking about adhesion. And so adhesion and invasion really end up, again, kind of going hand in hand for some pathogens. And we're going to spend the first of these lectures talking about adhesion. And I'm going to try and give a couple of generalized mechanisms that you'll see a lot of pathogens use. And these can kind of get extended to a lot of the pathogens that you guys are going to be thinking about. Starting next week. So next Monday, we're going to begin the process of begin the process of getting you guys into groups and you guys choosing pathogens and thinking about some stuff. And so that'll be all made clear in Monday's lecture. But I wanted to make sure that we cover good stuff. All right, so what are we going to talk about today? I'm going to talk about specifically mechanisms to study adherence, and I'm actually going to leave the methods to study adherence for the next lecture. But what is a roadmap for the next several lectures going to be? And I like to try and conceptualize things. I gave you a kind of a nice, cute picture that's in the Brock textbook talking about sort of adhesion and invasion and colonization and toxicity and things. And I like it because it's a good visual picture. But when thinking about things a little bit more conceptually, there's a different type of map that I like to draw in that we have exposure in those events, and in some ways it's part of this class, and in many ways I think it's obvious in a lot of ways how you become exposed to these things, contaminated food and so on. When we start talking about examples that we're going to see in the next couple of lectures, try and illustrate where are you contracting these things. And where we're going to spend a good amount of time today is talking about adherence to skin or mucosa. And this is where sort of rubber meets the road for pathogens. This is this initial interaction with host tissues. And as we'll see in Monday's lecture, invasion is sort of this next step for many pathogens. But there are a good number of pathogens that do not invade epithelial cells, okay? And that they essentially survive out in the wilderness, so to speak, of your gut and gut flora, as we'll talk about with many of the intestinal pathogens, or fending off the host immune system if you're a topical pathogen like Staph aureus, for example, and that you're essentially duking it out, looking for nutrients, and evading the immune system. And as we'll see in the next lecture, invasion of host cells is one really profound way to evade the immune system and acquire nutrients, right? And as we move forward in this section of lectures, we'll then begin to talk about where do exotoxins, toxins that are produced by bacterial pathogens, when do these come into play? And how are these, what are mechanisms targeted in host cells by toxins? And then ultimately these things lead to the disease presentation that we see for bacterial pathogens, right? So you're contracting salmonella, you ate some exposed food, those salmonella adhere to epithelial cells in the ileum of your small intestine. They're invading those epithelial cells and producing a host response that ultimately promotes the colonization of salmonella in your colon and ultimately leads to sort of basically inflammatory diarrhea, leading to the spread of this bacterium in populations. And that's non-typhoid salmonella in a nutshell. It doesn't produce any toxins, but then there are players like Vibrio cholera that causes cholera that makes a very nasty toxin that elicits a much stronger inflammatory and diuretic response. And so we'll start to see many of these different mechanisms in the coming lectures. But today we're going to start at the sort of the top of this set of terrible events for an infected individual, and thinking about adherence. And so there are a couple of different flavors of adherence. There's what you would term non-specific adherence to host tissues. And the sort of canonical example, the classic example I'm going to give is the formation of biofilms and essentially what that lifestyle does for pathogens. And then I'm going to take you through a couple of specific examples looking at essentially these extracellular protein structures produced by many pathogens used to adhere to specific host cell structures. We'll kind of focus on carbohydrates when we talk about pili. And then I'm going to take you through some examples of afimbrial adhesions, which are much more specific, intimate mechanisms of cellular adhesion that a lot of bacterial pathogens use. So, in terms of these specific or non-specific adhesion type mechanisms, I wanted to use this case of biofilms, in that they're important medically, and that they are able to form on abiotic or biotic surfaces. And I'll kind of illustrate what a biofilm is in just a second. But these characteristics I want you to kind of see and think about when we start talking about the structure and what's actually going on with them. And so these things forming on abiotic surfaces, this is important clinically in that there are many abiotic devices used clinically, right? So in, say, long-term hospital care, catheters, for example. That's an inanimate object that is introduced into the urinary tract of people in hospitals, right? And formation of biofilms on catheters is one of the major sources of urinary tract infections in hospitals. Implants, so having a joint replaced, for example. If you end up with staph bacteria that may end up on those types of devices, you can end up with essentially a very deep tissue infection because of biofilm that had formed on an abiotic surface. Yes?
So is this pre or post implant? Was the biofilm on there before it was inserted, or?
It could be, or that it may
on.
have formed on that thing. You had like a few bacteria that were on this device, put in your body, and they essentially grow on that device surface.
I'd hope they'd clean it before it inserted in your body.
I hope so. Yeah. But yeah, which is one of the reasons a lot of medical devices, why they are essentially most of the time these things are irradiated before they would ever even come near patients. Basically kill any bacteria that might be on them. So in addition to cells that are in these biofilms that we'll talk about, there are a number of things that are part of the composition of these large multicellular structures. And these are extracellular proteins that are secreted by the bacteria. There are extracellular polysaccharides for gram-negative bacteria. These include LPS, for example, and other polysaccharides, and extracellular DNA. And so in these biofilms that we're going to talk about, there's sort of the growth and death of bacteria. When bacteria die in lice, they liberate a ton of DNA. And there is some evidence now as well that there are some bacteria when they're growing in biofilm that they'll actually export DNA or soy seeds. And that as these large structures, and I say large, and that they're on the order of microns, like hundreds of microns in that they're visible to us, which for bacteria, and that's kind of a feat in and of itself, right? And that these structures, as they develop, begin to essentially have sort of multicellular properties in some cases, and they'll begin to contain channels for basically water and nutrients to fuse into parts of these biofilms, and that cell-cell communication is a major player. And so if you took microbiology with me, I spent a bit of time talking about quorum sensing. Essentially, this is a chemical method of communication between bacteria that's often mediated by compounds such as acylhomoserine lactones in gram-negative bacteria, or small polypeptides in gram-positive bacteria. And what these molecules are doing is essentially helping, or it's a way bacteria use to essentially sense population density. So if you're making a small amount of some chemical that can be smelt by everyone in the room, if there's one individual in a room, you won't smell too much. Now, at the population density of this room, everyone's fairly comfortable, right? Now, if we were to put 200 more people in this room and essentially, leave the lights off and turn off the air conditioning, the room might get kind of smelly. And that's essentially what's going on with bacteria. We would know that there are 200 people in here, even if the lights were off, and that, it would be really hot, really smelly after a while. And that's essentially more or less what bacteria are doing. They're using chemical communication to essentially sense and respond to the number of people or number of individuals in a population. And that, as we'll see when we are talking about some of these biofilm properties, is that one thing to keep in mind is that as these biofilms grow and develop, bacteria in them, they have differential metabolic activity. So they're not all seeing the same nutrients, they're not all eating the same things, and they're not all essentially experiencing the same world, right? So imagining this very, very crowded room, we had one window that was open. Those of you near the window would have fresh air. Those of you on the other side of the room would be not experiencing that fresh air, right? So these are just some things to keep in mind as we start talking about biofilms. So biofilms are multicellular structures that emerge from bacterial growth on solid surfaces. And so what the basic process of how these develop is that a small number of bacterial cells adhere reversibly to a surface. And so the small population, you might walk in the door, you might walk back out the door, right? And as these cells adhere and grow and divide on this surface, some stay, some go. Eventually, as the population gets larger and larger, these cells commit to life on this solid surface. So you can kind of think of, you're able to walk in and out of this door when there's only a few people in the room. If there's 200 people in this room and you walk in halfway into the room, it's going to be really hard to get back out, right? And so this is this idea that as these grow and this lifestyle becomes committed, this attachment to the surface is irreversible. And that these cells grow and divide on this solid surface, and all the while they're secreting protein, they're secreting exopolysaccharide. Some of these cells are dying and their DNA is spilling out. You have basically this big, sticky polymer matrix keeping all of these cells together. And this is during the stage of microcolony formation. So this is when cells are growing, they're dividing, and then ultimately these microcolonies kind of coalesce into a much larger structure, and this is this process known as maturation. And this is where really these cells are becoming highly differentiated in terms of their metabolic activity, what they're doing. Cells at the center of a maturing biofilm, they're experiencing less oxygen, they're experiencing much higher concentration of quorum-sensing molecules. So they're engaging in different behaviors than cells towards the outside of these biofilms. And then post-maturation, you end up with this very mature biofilm that essentially the exterior cells, they're still vegetative, they're still growing. These are being shut off. And I kind of label it a sixth step, this reinitiation. So this idea of, well, where did these bacteria come from? Frequently, they're coming from a biofilm from somewhere else, and ending up somewhere else, and basically initiating a new biofilm. So again, this extracellular matrix that's developing along with these growing cells is comprised of proteins, polysaccharides, DNA and RNA from lice cells. So you end up with this very tight, sticky web of extracellular material holding all of these cells together. And that cells deep in sort of the core of this biofilm. And I like this figure in that you can kind of think of these cells with having different colors being differentiated in terms of their metabolic function, what they're doing, what they're eating, as well as some additional properties in that they're sensing and responding to a much higher cell density than cells at the exterior of this biofilm. And so if we want to think about a property like motility, for example, very high cell densities in most bacteria is frequently a signal to stop being motile. So stop making flagella and stop basically swimming. Whereas low population densities often is a cue to engage in cellular motility. So you have this highly differentiated sort of lifestyle in biofilms, and it's actually not terribly unlike eukaryotic tissues, right? In that the tissues, epithelial tissues that we've talked about, there's epithelial cells covering connective tissue that's filled with lots of other different cell types. And so biofilm growth is actually an interesting sort of very primitive model for multicellular behavior, as well as being an important thing to study in terms of pathogenesis. So, as I mentioned before, what's in a biofilm? So it's composed of primarily water, and this is well over 90% of a biofilm. So essentially what a biofilm is a hydrogel matrix with bacterial cells in. And so bacteria make up between 2 and 5% of any given biofilm. The remaining portions are polysaccharides, proteins, and DNA and RNA from lice cells. And these compositions vary depending on bacterial species, but typically polysaccharides and proteins are the big components of biofilms. And so if we were to take any very matured, sort of all grown up biofilm and look at what's going on in it, if we were to look under a microscope, is that we would see that cells form clusters in these mature biofilms. That you see basically bundles of cells spread out through this very dense extracellular matrix. And ultimately, you end up having big voids emerge. And some of this is because of fermentation by some bacteria, which can release carbon dioxide gas, for example. You can end up with these sort of gas bubbles and voids in these biofilms, which can ultimately allow for channels to form. And these channels ultimately allow for a lot of nutrients to diffuse into these biofilms, and essentially promotes these sort of kind of distinct clusters of growth. And because of this thing being essentially a very sticky hydrogel matrix that's stuck onto a solid surface, these things are incredibly resistant to mechanical shear forces. And so thinking about your intestines, or thinking about your bladder, and your urinary tract, for example, these are high shear moving fluid environments. And that, especially for urinary tract infections, for example, biofilms are very, very serious. And one of the main reasons that you end up with sort of chronic urinary tract infections. And so the amount of fluid moving through a very fairly narrow diameter, right, is that the shear forces of urine leaving your body, or in your urethra, for example, are very, very high. And so E. coli that frequently colonize folks with urinary tract infections, like basically the movement of your urine is not sufficient to dislodge these types of biofilms because of the mechanical strength of these this type of growth. Now, there's some additional properties beyond just physical strength that make biofilms particularly nasty when it comes to basically colonizing us and being a lifestyle that promotes bacterial disease. In that, as I mentioned, the behavior of the bacteria in a biofilm are differentiated, right? And that's largely driven by access to nutrients. And if we think about kind of a blob of cells growing in a biofilm, is that those cells most proximal to nutrients, say out here in the environment, their metabolic activity is very high. They're growing, they're happy, they're doing things that these bacteria like to do. And as we move deeper and deeper into this biofilm, is that the metabolic activity of these bacteria and their growth rate slows. They become more and more dormant. If we were to look deep in the heart of some nasty biofilm, those bacteria essentially look like they're just dormant. They're not dead, they haven't lysed, potentially, but they're just sitting. They're not doing anything. They've got nothing to eat, but there's no sort of strong signal for them to die, or they haven't died yet, and they're hanging out there. And so this is a particularly nasty problem when we want to use antibiotics, in that many antibiotics function on vegetative cells. And so they're most effective when bacteria are growing and dividing. And so this is because many antibiotics, for example, that we use clinically are bacterial ribosome inhibitors. And so if you're metabolically inactive and you're dormant, you're being treated with a ribosomal translation inhibitor, you don't care, right? Because you're not making new proteins, you're not doing anything. You're just sitting and doing nothing. And so all of these cells that are actively making proteins, making enzymes, performing metabolic functions, growing and dividing, these folks die. These bacteria die during exposure to antibiotics. But these very dormant bacteria in the heart of a biofilm, essentially they survive this because they're essentially immune to antibiotic action. And so once treatment stops, we've essentially kept around this population of absolutely dormant cells. But now everyone that was consuming all the nutrients before it could get to them, they're all dead and gone. And now essentially they begin active vegetative growth and we establish the same basically large colony biofilm in a particular environment design. So, biofilms, very nasty, right? It all arises from a very non-specific adhesive event. There's no particular receptor or protein that bacteria have that's like, you know what, I have this extracellular protein and it's all about grabbing onto glass, right? I mean, there's not really a protein that you would want to have that's like silicon dioxide. That's what I want to bind, right? Or saying binding to stainless steel and doing this. It's not something that essentially bacteria are willfully wanting to hang out with. So that said, biofilm formation is the result of nonspecific adhesion that ultimately, because of growth and secretion of proteins and extracellular polysaccharides, essentially make bacteria stick irreversibly to both abiotic and biotic surfaces, and their continual growth on, say, a piece of glass, a piece of stainless steel, a piece of plastic that might be part of an implant, you name it, they'll happily grow on this. And it's also believed that this lifestyle, of growth is important for some pathogens that are basically aggressively colonizing your intestines, for example, during infection. So Vibrio cholera, for example, is believed to be forming biofilm on the surface of your epithelial cells during a cholera infection. So, how do biofilms protect pathogens during infection? I think I answered some of these in that bacterial biofilms, they're very resistant to antibiotics because a lot of the bacteria in a biofilm, they're not planktonic. So planktonic is essentially free-living growth, and that biofilms are very difficult to physically remove. So these shear forces in your intestines, in your urogenital tract, your nose, your mouth, these are all they don't seem like they're high shear forces. There are. So when we swallow food and it's moving through our GI tract, there are considerably high shear forces of that material moving through your intestines. And that biofilms are not easily dislodged by the forces exerted by fluid movement in our body. And actually, when we want to think about bacteria and sort of their interaction with antigen-presenting cells, for example. So essentially phagocytes and consuming bacteria. So we presented this model, we went through this model talking about adaptive immunity, where essentially phagocytes are eating bacteria, grinding them up, putting things onto MHC receptors, and communicating with T cells. That bacteria that are growing in biofilm are far less susceptible to phagocytosis. So this extracellular matrix that they've wrapped themselves in, this is not really promoting phagocytosis. And that essentially, by virtue of this type of growth, these bacteria have essentially put themselves on the same kind of measurement scale as the antigen-presenting cell. So no longer are you kind of a one micron diameter cell. Now, by virtue of growing in this biofilm, you are a 100 to 200 micron blob that an antigen-presenting cell is not going to be able to consume by phagocytosis. So, some pathogens that require biofilms to cause disease. So these are bacteria that cause a variety of different conditions, and that by virtue of our ability to study them in pure culture, what we found is that if we find genes, say, that are essential for the formation of biofilm of any one of these bacteria, we disrupt those genes and ask, did this affect virulence? Essentially, most of these bacteria, if I go and find those genes, knock them out, these bacteria become almost avirulent, that they are unable to infect animals. So the first of these is Legionella pneumophila, and this is the causative agent of Legionnaire's disease. And it's actually a really fascinating pathogen, and we'll talk about this bacteria again. We talk about effector proteins in terms of sort of intracellular lifestyles. And what this particular bacteria causes is Legionnaire's disease, which is an infection in your lungs. And what this infection is basically the cause of the disease phenotype is that this particular bacterium is very adept at invading alveolar macrophages. So there are macrophages that are swimming around in the mucosal fluid in your lungs, killing off all the stuff that essentially we breathe in, all kinds of nasty stuff, right? And these macrophages are essentially gobbling that stuff up and keeping our lungs a fairly sterile environment. And Legionella pneumophila is actually really, really good at invading these macrophages, not because it's a professional macrophage pathogen, but that its normal life cycle, where it normally lives and grows, is actually invading amoeba. And so its true host is unicellular amoeba, and these amoeba actually frequently grow in water towers. Remember from like a kind of high school biology class, I'm assuming some people have looked under a microscope and seen amoeba moving around in pond water, right? And so water towers actually have a ton of amoeba in them, which is fun. But so Legionella can propagate itself in these amoeba and ultimately give you Legionnaire's disease if that water is aerosolized and you breathe it in. The other is uropathogenic E. coli. I think its name hopefully says enough in that it is a urinary tract pathogen. It's the most common bad actor in urinary tract infections in both the developed and developing world. And one of the nasty things about this particular bug is that you think you've killed it off with antibiotic treatment, and 6 weeks later you have, again, an aggressive urinary tract infection. So clearing UPAC infections is really, really hard because of the formation of biofilm. Staphylococcus epidermis and Staph aureus. These are some of the most common nosocomial infections, so hospital-associated infections are because of these players. And that these are things that frequently contaminate catheters and plastic implants, in part because they are, as hopefully the species name here says, spells out for you, that they're normal inhabitants of our skin. So they like to hang out and live on your skin, and why we take such great care, or why doctors take such great care in trying to maintain an environment. And these are great friends of ours on our skin, but if you have lesions, you have basically these cells end up in the wrong place, and given a great place to grow on, like a piece of plastic, they become very, very nasty, opportunistic pathogens. Streptococci, so these are bacteria that frequently grow on your teeth and cause a number of periodontal diseases, as well as, say, Pseudomonas aeruginosa, which is a very famous lung pathogen for people who have cystic fibrosis. And so there's sort of this kind of classic green sputum that CF patients will have, and that green color is actually from a basically iron scavenging compound that's produced by P. aeruginosa, and also as an opportunistic pathogen for burns. So if I burn my arm, there's sort of a couple of actors that would typically end up causing a very nasty infection. So all of these are examples of bacteria that essentially, without the ability to form biofilm, they're almost avirulent bacteria. So it's a huge part of their virulence life cycle. So before we head into specific stuff, are there any non-specific questions?
I do.
Yes.
So with these surfaces that some of these abiotic pathogens are colonizing, where does the initial nutrition come for them to actually grow and replicate and be happy?
So if say this were a catheter, let's say, right, would be that I would be taking this out of some kind of packaging, administering this thing, and whether it be from me as the person administering a catheter, or from the skin of the person being infected, essentially, is that most of these pathogens that are, especially
Saltmongers.
these bottom four. Legionella is a very, very specific event. These bottom four, essentially, if I were to sequence, essentially my poop or my hands or whatever, look at the bacteria that are there, I would find these there. And so these are essentially diseases caused by bacteria who are ending up in the wrong place with a great environment to grow. And so the initial source of nutrients is essentially your bodily fluids, right? Or bodily fluids.
Right.
Yeah. Yes.
Okay, so the slime that's, they're secreting, I guess, are they secreting it, or is this just a product when so many bacteria get together? These are just waste products and other things that end up forming around them?
Both.
Okay, so it's,
I mean, it's just, they basically kind of turn into a big dumpster.
okay,
Yeah.
that's trash.
Yes.
Are all bacteria can form biofilms, or just are there more like chromosomes that form biofilms?
So can all bacteria form biofilms? The very sort of sweeping answer, yes. And that this seems to be and there are some very, very compelling arguments that the primary mode of growth for most bacteria, like at large, is this type of growth on solid surfaces and very tight association. And that's sort of our perspective that's been derived from looking at bacteria in isolation and in liquid culture and stuff, is that these are environments that they would never release. Now there are some aquatic bacteria, some marine bacteria that appear that for some time were thought to be, that they don't form biofilms, they just kind of are swimming around. So Vibrio cholera, for example, was thought to be sort of like this. But more rigorous kind of microscopic analysis of where are these Vibrio species in marine environments, you actually find them living on small particles in ocean water, for example. So they really like to grow on chitin, for example, which so basically like crustacean shells and stuff are made of predominantly chitin. So as these animals die and whatnot, and their sort of bodily material ends up in ocean water, these bacteria are essentially growing on that and eating it. So yeah. So there's a long answer for a yes or no question, but by and large, most bacteria appear to grow in biofilm. Okay. Yes?
So whenever a patient will take an antibiotic for, let's say, that third disease, staph illness or whatever.
Yep.
So the antibiotic likes to a high metabolic activity.
Mm-hmm.
Does the antibiotic take away the high activity, intermediate, and low activity, and then the dormant is still there?
Yep.
Does that become activity? It's not dormant anymore, and that's how the virus or whatever bacteria keep growing?
Yep. Yeah. So those dormant bacteria are largely unaffected by toxic stuff. And so what's that?
That's why it's important to finish.
Yes. And so if you are given antibiotics, finish the course of your antibiotics. And that, even in the presence of drugs, as those more active cells die and essentially go away, those dormant cells are now going to be experiencing nutrients, and those same drugs are going to be around. So they'll start eating and growing and then be killed off. by those drugs that are still around. So, so those 10 to 14 days while you feel better, that's why there's a lot of arguments to finish courses of antibiotics. So, great questions from both of you guys. All right, so there are obviously like other modes of non-specific adhesion, but this idea of biofilm growth, I think, is the most pertinent for bacterial pathogenesis. What I want to move on to is talking about some very specific mechanisms of adherence. And what I hope we see when we kind of like look at what some of these mechanisms are doing is that, yes, they are very specific, but in many ways they kind of feed back into this non-specific idea that it's this initial adhesion event, and you're going to, like, as a bacterium, grow and divide, hopefully in a particular environment. But I think it's kind of good to decouple these and to think about kind of very specific mechanisms versus this sort of broad idea of showing up and being kind of a organismal squatter in a host, right? Which is basically what you're doing in biofilm growth. So the first mechanism of specific adherence that I wanted to cover is through the use of an extracellular protein structure that many bacteria make called pili, or is the plural for pilus. And these are sticky, rod-shaped structures that extend from a bacterial surface. So there are some very kind of famous pictures that you can search and find online. These are E. coli cells that are producing fimbriae. And so you'll see the word pilus and fimbriae, or pili and fimbriae, used more or less interchangeably in a lot of literature for the context of this class. I'm not going to be really particular about differentiating them, and that they are, at least under a microscope, they're structurally similar, although there are some very striking differences between different types of pili and fimbriae. And the important bit here is that this tip of these long proteinaceous projections, these pili that are sticking out of the cell, is that the tip of these particular extracellular protein apparatuses often bind to a very specific host cell receptor. And that receptor is not necessarily a protein for pili, and it's actually a carbohydrate. And so most pili, for example, really like to bind mannose. And so mannose is something that is a sugar that's part of the extracellular matrix around epithelial cells. And it's a great, kind of very nice carbohydrate to specifically pick out of a mixture of different carbohydrates for these bacterial pili. And that they're often required for biofilm formation as well as specific adhesion. So these proteinaceous structures, they're actually able to bind to each other very well. And so those tips are good at binding carbohydrates, and the sort of proteinaceous parts are very good at sticking to each other. And so looking at these two bacteria that are making pili, you can imagine these proteins kind of weaving together and forming a very tight network between cells. Yep.
So you have the different fins, like say fin H is to the mannose, right?
Mm-hmm.
So they would have something different if it was like fin something else.
Yep.
It would attach. Okay.
A lot of them.
Not all of them.
Not all of them bind to mannose, but a large number of them do. Yeah. So, some examples of pili and pathogenic bacteria. So I kind of gave UPAC as an example for biofilm formation, and so kind of no surprise that they are also kind of prolific makers of pili. So we're going to go through a few different types of pili. So I'm going to take you through two gram-negative examples, so the type 1 pilus and the type 5 pilus of Neisseria species. So you remember Neisseria from our paper discussion, right? And then I'm going to take you through a gram-positive example in Corynebacterium diphtheriae, which is a very nasty pathogen that we now have basically kind of taken care of with vaccination. So, UPEC. So this is a gram-negative rod. It's essentially the E. coli that we work with in the lab with some extra nasty genes encoding a lot of these fimbriae and some other things that we'll talk about later on. And so this is the primary cause of community-acquired urinary tract infections, particularly in women. And it's found in the colon, but can travel to the urethra, and it's essentially kind of everywhere. And pili and biofilm formation, these are the major mechanisms of how this particular bacterium is engaging in its sort of pathogenic lifestyle. So showing up, binding to epithelial cells, quickly growing, establishing this biofilm on the surface of epithelial cells protects these bacteria from phagocytosis and the high shear forces of essentially going pee, and that they colonize the urethra. And remember that a lot of the things that bacteria, especially gram-negative bacteria, have on the outside of the cell. So some of these proteins, some of these pili proteins, for example, lipopolysaccharide, DNA from lice cells, and some other compounds that we talked about. Remember that these are antagonists for the innate immune system, right? So these bacteria showing up and growing on the surface of epithelial cells. It's not as though these epithelial cells are saying, man, there might be something out there. No, they know what's out there. And this type of growth on bodily surfaces or epithelial surfaces, this is a very strong activator of the innate immune response. And so it's part of the inflammatory response that you get in a urinary tract infection. So what I'm showing here are a number of kind of like electron micrographs up here of a essentially UPEC biofilm that is formed on the surface of epithelial cells in a mouse urethra. These are very close-up images of the surface of this biofilm. That you can kind of see the tight network of cells, and that these biofilms ultimately kind of are sort of push their way between epithelial cells. You're seeing some of these images, and so it's a great infection model for understanding biofilm growth and its connection to pathogenicity. And I feel bad for the mice that gave us these nice pictures, but so they specifically adhere to the epithelial cells in the urogenital tract, and they do this using type 1 pili. And so these type 1 pili, as we'll kind of structurally look at, these bind to mannose on host cells. And so this is part of the extracellular matrix of epithelial cells. And mannose is a very prominent sugar. And that these types of pili, because they bind mannose is also very prevalent on the surface of red blood cells. And so these particular type of pili can cause something known as hemagglutination. And so this is aggregation of red blood cells. There's another type of pili that are also produced by UPEC. For example, the P pili, they don't cause hemagglutination because that particular pilus does not bind to mannose. And so they have different receptors. A lot of pili bind to mannose, and by virtue of that, they will induce hemagglutination if they see red blood cells. And these are assembled on the outer membrane by a pathway known as the chaperone-Usher pathway. So that's a pathway that I covered when we talked about protein secretion systems a few lectures back. And the way that this particular type of pilus works is that it is assembled on the outer membrane of gram-negative bacteria that make type 1 pili. And what happens is that these proteins that ultimately end up as this pilus, they're secreted by the sac system. They're bound by a chaperone that basically helps this subunit fold. And this chaperone delivers these subunits to this outer membrane pore, and these subunits are added onto this growing pilus rod, and then essentially these chaperones cycle around and continue building this structure. The important thing is that those tip proteins, say fin H, for example, that is the first protein that's delivered to this pore, and essentially is the initiating nucleus for the formation of these following subunits. So essentially, tip, adapter proteins, and then major subunits that get added on to essentially grow this long proteinaceous structure. So there's lots of different types of pili. I'm trying to give you an overview of kind of the major types that you will see in pathogens. And the next example I'm going to take you through is the type 4 pilus, and this is essential for the adhesion of a pathogen, Neisseria gonorrhoeae. And so this is the causative agent of gonorrhea, the gram-negative diplococcus. We talked about this in the last lecture, and that they like to grow in this diplococcus arrangement. So it's essentially two spheres smushed together. This is an electron micrograph of Neisseria bacteria on a solid surface. And so it's a cause of gonorrhea in adult humans. And if you are a carrier of Neisseria gonorrhea as a woman and you give birth to a child, it can lead to gonorrhea eye infections in children. And one of the nasty things about this particular bacteria is that it's often asymptomatic. And so there's a lot of asymptomatic carriers for this particular pathogen. And this type 4 pilus that we're going to talk about, this is critically important for the colonization, basically adherence and colonization of the urinary tract by this particular bacteria. And this is mediated through a family of, also we'll talk about these OPA proteins. They're afimbrial adhesins. We'll talk about those at the end of today's lecture. But we'll first start talking about the type 4 pilus. So they're found in a lot of different gram-negative bacteria. Essentially what this is, unlike the chaperone-Usher pathway that we just talked about, this is a modified type 2 secretion system. So if you remember from the secretion system lecture, essentially the type 2 secretion system, there's an outer membrane pore, there's kind of an intermembrane spanning adapter, and then all of the subunits that are going to get excreted through this particular apparatus are delivered to the periplasm via the sac system, and this will become clear in a second. This is really important for adhesion to host cells, bacterial aggregation, and a type of motility known as twitching motility. And I don't think I have any videos. Anyhow, if I don't, I'll put some up on the site. And unlike the type 1 fimbriae, which that whole assembly event is happening on the outer membrane, this structure is essentially assembled on the inner membrane. And I'll take you through what goes on with this. Okay, so these pili subunits, these are secreted into the periplasm via the sac system. And they actually have a membrane binding component, that part of them are an integral membrane protein, and so they reside essentially on this periplasmic face of the cytoplasmic membrane. And what happens is that these subunits diffuse into this type 2 secretion structure and are sort of ratcheted into place by the action of an ATP-dependent rotary motor. It's somewhat similar to what, say, part of this is similar to the F1, F0 ATP synthase, for example. And so it's this ATP-dependent ratcheting motor. And so these subunits diffuse in, and elongation, there's one ATP motor that's responsible for elongation. And so this ratchets these subunits together, makes this long projection. And the really phenomenal thing about this, unlike what we saw for the type 1 fimbriae, which is essentially I'm going to build this long structure, and they're just going to kind of hang out here and hopefully bind some stuff, and that'll be great. What this thing does, actually, is that essentially this pilus is built by one ATP rotary motor that connects to this apparatus. Then for retraction, a separate rotary motor connects and burns ATP and essentially ratchets this whole structure the other way and spits these subunits back out and allows for retraction of this protein appendage. And so there's two separate rotary motors that connect to this apparatus, and the switching between these motors is thought to have to do with the mechanical forces that this pilus is being built and elongated and eventually sticks to a surface, and that sticking to a surface causes mechanical strain on this structure. And that mechanical strain is thought to change the structure of this pil C and pil M subunits, favoring docking of one ATP motor versus the other. So there's a lot of cool physics involved in this particular system beyond the scope of this course. But the important thing about this particular pilus is that it's able to project a protein appendage. And essentially, it's like kind of like throwing out a grappling hook, and this bacteria is essentially pulling itself to where that particular attachment event happens. So, as I was kind of describing, these pilin monomers, and so this is the inner membrane. You can imagine this thing projecting through this outer membrane core, is that this pilus is being extended, attaches, and this attachment event is believed to be a physical cue to switch motors and essentially begin retraction of these cells to where that attachment event happens. And so assembling this tip protein goes out first, and it's sort of the nucleator for all of these major subunit proteins that get added on afterwards. So, are there any questions about this?
So they diffuse back through and get assembled on the outside here, and then get pushed back? I was looking at go to the slide where it shows the other assembly.
Mm-hmm.
And it shows the pill A or file A, I don't know what you call it. Well, it shows them embedded here in this membrane, so they have to get back through here and then go back through?
So this whole structure gets built from the bottom out through this port. And then when this is retracted, these subunits basically go back into the cytoplasmic membrane. And so what can end up happening is that you're using the same subunits over and over and over again. If this thing gets out, pulled back in, out, pulled back in.
Because I was just confused about these green things down there.
Yeah, and so these are where essentially the energy.
Okay.
These things dock onto this pil C protein and essentially ratchet this whole structure around. I was at a meeting a couple of weeks ago, and some people have done some really insane, like cryo-EM microscopy, like looking at this whole process. And I've been trying to find their movie so I can put it up for you guys. Right? It was totally nuts. But it's a really awesome structure, and I'll post some videos of twitching motility. So essentially, bacteria throwing out these proteins and essentially pulling themselves across solid surfaces using this sort of a mechanism. Okay, so I've taken you through 2 gram-negative versions of pili, and it would be remiss to not talk about the gram-positive version of these structures. And it's fundamentally different, largely because, remember, protein secretion for gram-negative versus gram-positive bacteria, it's an entirely different beast, right? Having an outer membrane places some very important constraints on gram-negative bacteria about displaying proteins versus gram-positive bacteria, which having that cell wall, you have to use that as the anchor for these extracellular protein structures. So the canonical example for thinking about extracellular pili in gram-positive bacteria is Corynebacterium diphtheriae. So it's the causative agent of diphtheria. So this is a very nasty, deadly disease that's now curtailed by the use of vaccines. So it's part of a combinatorial vaccine given to children now, so that children essentially don't get growth of this particular bacteria in your throat, which is a very nasty picture of essentially what this bacteria does. It adheres, begins growing, and makes this pseudo-membranous biofilm structure in the back of your throat, growing over your tonsils, and essentially results in sort of this like nasty cough that I used to play a video of somebody with essentially a diphtheria cough, and it's probably this, and I'm not going to subject you guys to it, but you can look it up if you want to. But essentially, it's this like horrible cough, croup essentially, and essentially this pseudo-membranous thing gets big enough and nasty enough that essentially you can suffocate in some cases, but it also ultimately becomes a systemic infection and is very, very bad. So anyway, there's a vaccine for this, and it's important that we used it. So pili produced by this bacteria are very important for its adhesion to your upper airway, and these are actually physically attached to the cell wall of the bacteria. So it's composed of 3 proteins, SPA-A, SPA-B, and SPA-C, so the SPA-type pili, and that, like most all gram-positive extracellular protein structures that are made, that are secreted, these require sortase enzymes. And so you remember sortases are essentially these shuttles that take secreted proteins that have specific protein motifs in them. So this LPXTG is a very common sortase motif in proteins. These get bound, added onto specific lipids in some cases, or the sortase basically carries these proteins around and essentially allows for covalent bond transfer between this sortase SPA-A complex, for example, to form a larger structure. And eventually, a housekeeping sortase is responsible for moving this entire extra big cellular protein structure onto the cell wall. And so shown here is the SPA pilus of Corynebacterium diphtheriae. And so this essentially SPA-C, SPA-A, these things get covalently bonded together. More and more SPA-A subunits get added on. And finally, there's a terminal SPA-B protein added on the end, and then this is what gets anchored to the cell wall. And so this tip protein, this sort of major subunit, and then there's a terminal subunit that's essentially responsible for getting anchored to the cell wall. And so the SPA-A protein is the most abundant, and essentially provides this long length for these pili. And so they're structurally different proteins that, if you're staining and looking under a microscope, they're going to look very similar to the gram-negative proteins that we just saw. But they're functionally they perform the same function, but in terms of protein structure, they're very different proteins. All right, so we're to think about this particular table here, right? So how are these proteins secreted across the cytoplasmic membrane? In all cases that we saw, they had to be secreted by the general secretion system, right? So those proteins get out into either the periplasm or the extracellular environment for gram-positive bacteria through the sex system. So where are these pili assembled? So type 1, remember this is on the outer membrane. So the assembly event happens on the outer membrane. For type 4, the assembly happens on the cytoplasmic membrane. Whereas the SPA type, it's outside the cell. And so it's happening in the extracellular environment and being added onto the cell wall. So the order of assembly is, if you remember, these type 1, that there was a tip, there's kind of an adapter protein, and then the major protein subunit. Type 4 is essentially the tip protein, followed by all of the major subunit proteins. And these SPA type proteins, if there's a tip, a major subunit, and then essentially an anchoring subunit to put that onto the cell wall. Okay, so in the last 5 minutes, we'll see where we can get thinking about afibrillar adhesions, though we can wrap this up briefly in Monday's lecture. So, afibrillar adhesions are, as they sound, they're not pili, they're not fibrae, they're not long proteinaceous appendages, and these are actually far more specific than pili. And that we're going to go back to our example of Neisseria gonorrhoeae, and that this uses one type of appendage to essentially make a long-range interaction that then has an afibrillar adhesin protein that mediates a very, very specific interaction with a particular type of host epithelial cell. And so they have this essentially OPA proteins that anchorage happens because of type 4 pilus throwing out kind of molecular grappling hooks, drags into a particular host cell type, and is able to anchor itself to these CCAM receptors. So what these receptors do, it's not important for what we're talking about in this class, but adhesion to very specific types of epithelial cell proteins is what I want you to take away from these afibrillar adhesins. And so what are they? What are they most typically? They're most typically type 5 secretion systems. Okay, so if you remember back to our secretion system discussion, that these are proteins that get secreted out into the periplasm for gram-negative bacteria and essentially are put into the outer membrane by the beta barrel assembly machinery, and that they often have a passenger domain that essentially gets secreted out through this pore. And so what I'm showing here are some type 5 secretion systems from a pathogen, Bordetella pertussis, which is the causative agent of pertussis. It's a nasty cough, whooping cough. This is kind of the common name for it. And what goes on is that these particular type 5 secretion systems, they are projecting out a very specific protein tip that binds to a specific host surface receptor. And so there are two that are very common in most Bordetella species. So there is FHA, filamentous hemagglutinin, and another one, pertussin or protactin. And both of these bind to a couple of different host cell surface receptors. And so it makes fimbriae to essentially establish initial interactions, and then uses these type 5 secretion systems to mediate very specific high energy interactions with host cells. Essentially locks these bacteria onto very specific types of, in the case of Bordetella, ciliated epithelial cells. And so, to summarize today, these bacterial factors involved, so the biofilm, your pili, exopolysaccharides, extracellular DNA, essentially make a big sticky mess for bacteria to stick to stuff. Specific adherence, whether this be fimbriae or there are fimbriae, afimbrial adhesins, and these pili, they're usually binding to carbohydrates. Afimbrial adhesins, these are typically binding to membrane proteins or glycosylated membrane proteins. And so pili, these interactions are very weak, whereas afimbrial adhesins, these interactions are very, very strong. They're very, very specific. And so pili, they're really helping to resist shear forces, for example, in the body. And these same things are happening with afimbrial adhesins. But what we're going to see in the coming lecture is that these are very important for invasion. Okay? And so that is where I want to leave today. I'll see you guys on Monday.